After the main chart of the cell injury, mechanism of cell injury, we are now going to see what will happen if there is mitochondrial injury. There will be failure of ATP generation. First step, there are 3 points. First point is failure of ATP generation. Why? If there will be mitochondrial injury, I have written mitochondrial injury in short form as MI. In the mitochondrial injury, there will be loss of mitochondrial membrane. So there will be failure of normal oxidative phosphorylation that leads to decrease in the ATP, and decrease in ATP will follow the same pathway of the chart and leads to nuclear cellular changes and serum enzyme estimation. The final result will be nuclear changes, cellular changes, and the SGOT, SGPT, serum enzyme estimation. The first point is failure of ATP generation. Due to mitochondrial injury, there will be loss of mitochondrial membrane. They will lead to the failure of normal oxidative phosphorylation. This oxidative phosphorylation generally leads to ATP formation. If there is failure of oxidative phosphorylation, there will be decrease in ATP synthesis that will lead to all the chart I just told you in the previous video. Now there will be formation of reactive oxygen species. If there will be mitochondrial injury, that leads to failure of oxidative phosphorylation, and there will be also abnormal oxidative phosphorylation that will lead to increase in reactive oxygen species or free radicals that will lead to deleterious effect. There will be damage to cell membrane, there will be damage to intracellular content, extracellular contents, that's known as deleterious effect. There will be release of cytochrome C that leads to apoptosis. We'll start in next chapter the apoptosis. In the cellular death, there are two mechanisms, apoptosis and necrosis. In the next chapter we'll see. In mitochondrial injury, there will be increased permeability of mitochondrial membrane. If there is injury, we have seen there will be membrane damage, there will be liberation of intracellular enzyme. There will be increased permeability of mitochondrial membrane that will lead to leakage of cytochrome C, which is generally present inside the inner cell of mitochondria. Inner cell and outer cell. There is inner cell, there is outer cell. In between the inner and outer, there is present of cytochrome C. If there is membrane damage, there will be leakage of cytochrome C into the cytoplasm that leads to the apoptosis. In first and second point, the failure of ATP generation due to loss of membrane and the formation of reactive oxygen species, they both lead to necrosis, a pathway of cell death. But in the third step, in the if there is a release of cytochrome C into the cytoplasm, there will be apoptosis. So first and second point leads to necrosis, and the third point, this release of cytochrome C leads to apoptosis. We will now see or review all the things. See, cellular adaptations are generally reversible adaptation due to the stress put on the cell. There will be structural changes as a response to the stress. This allows the cell to survive and continue its function. In hyperplasia, there is increase in cell. This is the diagram. There will be increased size of organ. Mechanism in increased production of growth factor. We have seen the physiologic and pathologic examples in the previous video. In hypertrophy, there will be increase in size of the cell due to increase in cellular protein. It's not a swelling. There are also physiological and pathological example. In atrophy, there will be decrease in size and decrease in number, but mostly it is decrease in size because trophy means size. Atrophy means decrease in size. The mechanism is reduction in cellular organelles that leads to decrease in size. In metaplasia, there is conversion of one mature cell type into another mature cell type. What is the mechanism? Reprogramming of stem cell that leads to squamous metaplasia or columnar metaplasia. We have seen in previous videos. In dysplasia, there is disordered cellular development characterized by cellular cytologic changes. These are the 7 cytologic changes. If it covers all the layers, it will be known as carcinoma in situ or precursor of the cancer. The mechanism of cell injury, it's the most important cause of cell injury is ischemia or hypoxia that leads to decrease in cellular ATP generation, that leads to decrease in synthesis of membrane phospholipid, increase in cytosolic calcium that leads to reperfusion of calcium into mitochondria that leads to increase intracellular cytosolic calcium level that activate phospholipase and protease that leads to loss of membrane phospholipid lipid breakdown products. If phospholipid break, it forms lipid breakdown products. If protease activation, it leads to cytoskeletal injury, and all these steps leads to membrane damage. Here toxioxygen free radical leads to lipid peroxidation DNA damage that leads to membrane damage that leads to liberation of all the intracellular enzyme. As results, if there are seeing the nuclear, cellular and serum enzyme estimation, these are the final products of the cell injury.